Welcome to AJP Audio for March 2022. I'm Aaron Van Dorn. Dr. Giovanna Punzi and Dr. Weinberger are researchers at the Lieber Institute for Brain Development in Baltimore, Maryland, and authors of a new article in the March issue of the American Journal of Psychiatry looking at the genetics and brain transcriptomics of completed suicide. They'll discuss their findings looking into the difference between those who die by violent and less violent means of suicide. Afterwards, we'll check in once again with Editor-in-Chief Ned Kalin to find out more about the March issue of AJP. Dr. Weinberger, your study looked at the brains of people who died by suicide, both by violent and non-violent methods, by looking at transcriptomic and genomic features, as well as neurotypical patients. What did you find?
Well, we were very interested in the question of people who die by suicide. Is this one condition, or is the manner by which people die by suicide important in understanding the biology of suicide? So working with, I'm very fortunate to have had a postdoc, Giovanna Punzi, who's on this call. She's now a research scientist at the Lieber Institute for Brain Development. We had access to hundreds of brains of people who died by suicide. And it allowed us to ask the question of whether at the biological level, and by that I mean how genes are turned on and off in the brain of a deceased person, and at the genetic level, meaning how variation in the genomes of individuals who die by suicide may represent their risk for psychiatric illness. We were able to ask how these two biological determinants might differ based on the manner by which someone died of suicide. The assumption was that this manner may make a difference. And the basic finding of this study is that people who die by suicide using a violent means for suicide are biologically distinct, both in terms of the genes that are turned on in their brain at death and in terms of the genetic risk for major psychiatric illnesses. They are distinct from individuals who die by suicide using other methods. That is the overarching result of this study. And when we've identified maybe some very specific signaling features in the tissue that may explain these differences.
Dr. Punzi, turning to you, you found that patients who died by violent means differed from those who died by suicide by non-violent means. Can you tell us about what made these two categories distinct and what you found by looking at them?
We found that patients who died by suicide using a violent method were somehow different from other patients, including non-suicide, but also suicide by means that were non-violent. And that was both in terms of gene expression in the dorsolateral prefrontal cortex and in genomic profile of risk. So the differential expressed genes associated with suicide by violent method, in contrast to other patients, were they suicide or not, pointed to G protein-coupled purinergic signaling in microglia. Additionally, WGCNA, so network analysis, revealed that genes in the signaling associated with suicide by violent method were co-expressed in the context of mitochondrial metabolic activation. And I should point out that this data was unique to suicide by violent means. So it was unique in terms that it was true compared to patients, but also to controls.
In the paper, you mentioned that aggression was a factor in violent suicide deaths, but a lot of people might think, well, but suicide is kind of by its nature an aggressive act. What did you find? How does that differentiate between the two categories of people who died by violent and non-violent forms of suicide?
Yes, definitely, I agree with you. So of course, a suicide is always aggressive, in fact, and suicide by definition is a violent act. However, we wanted to focus on what's behind suicide when suicide is actually happening. So looking at how much violent it was at that moment on a scale. And that's because we believe that this would reflect a different state of mind. And this in turn might translate into a different biology fixed in the brain at the time of death. And indeed, we did have previous evidence of a link between suicide and aggression. In a previous publication, we found that a gene, and this gene is actually in the same network of the purinergic genes, was associated with suicide by violent means and with behavioral measures of aggression, and also with different brain activity during an fMRI task. So in this regard, I think in this paper, I think it's rather remarkable that the signaling that we found associated with suicide by violent means emerged also in a GWAS, in a genome-wide association study of a Drosophila aggression model.
What are the genetic factors that differentiated the patient groups?
In the paper, we have patients affected with schizophrenia, major depression, or bipolar disorder. So The patients affected with schizophrenia or depression who died by suicide by violent means were less genetically susceptible to their own disorder. So meaning that the GRS for the disorder they had been diagnosed with was relatively lower than that of other patients with the same diagnosis, but who didn't die by suicide or who chose a non-violent method. And interestingly, in the case of schizophrenia, the risk profile of suicide by violent means indicated even a higher genetic resilience to schizophrenia than other patients. We also used the GRS for IQ as a proxy of cognition and found that suicide by violent means were more genetically inclined to higher IQ than other patients. In the bipolar subgroup, in the presence of high IQ GRS, suicide by violent means had lower bipolar GRS compared with other patients, which is consistent with the other diagnosis. And then finally, interestingly, in the genomic profile of suicide by violent means was also not associated, which is surprising, with suicide attempt GRS, which in fact complies with the notion that suicide attempt may not catch the genetic architecture of the actual completed behavior, especially by violent method.
I think that's one of the really important observations of this study, which is that people who attempt suicide don't necessarily overlap biologically with the people who committed suicide. This is particularly true if they use violent means to kill themselves. You know, we have traditionally treated suicide as a homogeneous state. And these data, both genetically and at the level of genes that are on and off in a brain at the time of death, suggest that these are really multifaceted syndromes. And that when one uses a violent method to kill oneself, they are in a different group of people biologically than individuals who commit suicide using other methods. This opens up many areas of thought and future investigation for what is this telling us about these individuals and their state of mind and their emotional state.
Aren't these conclusions kind of counterintuitive from what you'd expect? The idea that people who choose a violent means of suicide would have less underlying expression of conditions such as schizophrenia or depression than people who died by suicide by nonviolent means.
Yes, it's a surprising finding. And it's a finding pointing to the fact that suicide by violent means may be a separate condition. In a way, these data are telling us that suicide by violent methods may be genetically inclined to less diseased condition. I don't know if that sounds good in English. And they might be cognitively have higher performance. So in a way, they just look genetically, in theory, as far as their genetics, they look healthier than other patients. And that's interesting to me, I think, because it has been speculated that mental disorders may actually be protective against suicide from an evolutionary standpoint. And that suicide may be the byproduct of a more developed brain.
You know, one of the things to remember about suicide by violent methods, in contrast to many other means that people have used, is you sort of can't change your mind. You can't undo it. This is, you know, it's an extreme mental state. No possibility of changing one's mind. So the mindset of somebody who hangs themselves or shoots themselves or jumps in front of a train is a very different mindset from somebody who takes pills or sits in a car with the engine running, where there's a long period of time where they still can contemplate the act that they're engaged in. And the suggestion is that these are not the same mental states. And they're not the same mental states either in terms of how the brain manifests this mental state at a molecular level. And they're not the same mental state in terms of people's genetic risk to have depression, schizophrenia, or bipolar disorder, which are the principal disorders associated with risk for suicide. And it also suggests that individuals who use these violent methods are not people that seem to be genetically like people who make suicide attempts, whereas individuals who commit suicide using other methods more passive methods are like the people, more genetically like the people that are make suicide attempts. So again, all of these data together argue that there is something unique at a biological level, both in terms of brain biology and genetic risk for psychiatric disorders in individuals that have chosen this violent method for killing themselves compared to the other populations of people who have committed suicide. Giovanna, is that consistent with the way you look at this?
That is also consistent with the data that we see that suicide by violent means have lower GRS for suicide attempt, because by definition, suicide attempters survived. And instead, we're looking at a phenotype where the method chosen is more lethal and so more effective. And indeed, we're looking at completed suicide.
What are the clinical implications for the treatment of people who are at risk of suicide?
At this point, we can only speculate. We can't really say based on just this study, but the association of the purinergic signaling with suicide is a novel observation. And so if this association, for example, if the association is also mirrored by peripheral biomarkers, then those may in future potentially differentiate patients who may likely shift from considering that to actually forcing it. So that will provide novel and more precise targets for therapy in short-term risk for suicide. But again, I think important implication is that suicide by violent means probably should be addressed as a separate condition.
What are the next steps for your research?
So what we find is that we link suicide by violent means with increased purinergic signaling in the context of mitochondrial metabolic activation. And the expression of these purinergic genes is enriched in microglia. So that might suggest a possible alteration in neuronal microglia communication that may be detrimental to brain function. In fact, microglia is supposed to modulate neuronal activation. So, but this data, so these findings come from a study on gene expression measured in bulk tissue. We cannot really say which cell types express these genes. So a next step, that step definitely will be to perform single nuclei RNA-seq and to look for differences in transcription at the single cell level.
Dr. Punzi, Dr. Weinberger, thank you for joining us today.
Thank you.
Next up, Dr. Ned Kalin on the March issue. Dr. Kalin, welcome back to another episode of AJP Audio.
Thank you, Aaron. It's good to be here.
The March issue of the American Journal of Psychiatry centers around insights into the genomic underpinnings of psychopathology. Earlier in this episode, I spoke with Drs. Weinberger and Punzi about their paper in the issue, but what else is in the issue?
So this issue is really, I think, a special issue because all of the papers are focused around how genetic variation is related to the development of different types of psychopathology and different types of illnesses. And more specifically, you talk with folks related to the genetics related to violent suicide, but also we have papers related to the genetics that underlie obsessive compulsive disorder, autism spectrum disorder, and some interesting papers related to rare genetic variation as it relates to the development of psychosis. And so it's a really nice compendium of papers focused on these different areas and bringing our readership up to date with new findings. And then also we have two overviews or reviews focused on really helping to make accessible principles of genetics that are particularly relevant to understanding psychiatry and psychiatric illnesses. One of those is a review by Dr. Francis McMahon, who is an expert in this area from NIMH. And he really talks about current methodological strategies to understand genetics, as well as sort of the state of the art from the standpoint of our findings, and also talks about, when we'll be able to think about what we need to do to be able to think about how we can use these findings clinically. And then in another review, we have a focus on how rare neurodevelopmental problems that are attributed to one gene, that is where there's one gene that's mutated, that results in a developmental problem that is both physical and frequently mental, how that can inform our understanding of psychopathology. This is by basically a group that's called the Genes to Mental Health Network, which is a group that's funded by the National Institute of Mental Health and NICHD. This review talks about rare genetic problems and their subsequent psychiatric symptoms and sequelae, and also provides insights into how we can better understand genetic underpinnings of psychopathology, because in these cases, we know that there's specifically one mutation in one gene that is having a very big effect on the likelihood of developing, in one of the papers that's presented in this issue, for example, in psychosis or schizophrenia-like symptoms. So we have this broad range of papers, and each are addressing different issues related to genetics, providing new insights. So let me get into a little more detail related to the specifics of the original research papers that we've included. I just gave you a bit of an overview of the reviews, which really set the stage for our readership and will serve as a means to hopefully make all of this a little bit more accessible to those that aren't doing this kind of work every day. But one of the first papers that we've included is a paper that looks at individuals that have this specific mutation called 22q11.11.2 deletion syndrome. And basically what this is a deletion on chromosome 22, which results in physical problems, some heart-related problems frequently, but also roughly a third of the individuals that have this genetic mutation will later go on to develop psychotic symptoms or psychotic types of disorders. And because of this relation between this one mutation and the likelihood of developing psychotic symptoms, this now becomes if you will, a genetic model to understand how this particular genetic alteration may be related to psychosis and to other types of psychopathology. What the investigators did in this case was look at a neurophysiological marker, which is called gamma oscillatory activity. And these are basically brain waves that represent activation in a variety of regions. And they're particularly important because they're thought to have a lot to do with the communication between brain regions at a sort of a longer distance across the brain. It's well known that these oscillations are dampened or reduced in individuals with schizophrenia. And there's an understanding also of, at the cellular level, of what the types of cells in the cortex are that actually contribute to these types of brain oscillations. And that kind of work has actually been pursued and has given us some insights perhaps into what goes wrong in individuals with schizophrenia. But what's interesting about this paper is that these investigators have demonstrated that individuals that have this particular genetic mutation that really don't have a primary psychiatric disorder but have a neurodevelopmental problem and then are likely to go on to develop psychotic symptoms, have reductions also in the same marker that we see in schizophrenic patients, which is this reduction in gamma oscillations. But also what's really interesting and important about this is that we know that this is related to this particular genetic alteration. So this provides some insights perhaps into the underlying genetic underpinnings of a critical neurophysiological change that's associated with schizophrenia. So not only does it provide insight into individuals that have this problem with this mutation, but it also, using that data, gives us some ideas about how to think more deeply about how genetic alterations may relate to the development of schizophrenia and some of its neurophysiological underpinnings. Another paper that's quite interesting is by Mahjani and coworkers. And this paper is focused on further understanding the genetic underpinnings or architecture related to obsessive compulsive disorder. And this is an interesting paper because obsessive compulsive disorder is an illness that also we think of as being neurodevelopmental to some extent. It occurs in roughly 2% of the population. And there have been a number of studies that have tried to look at the genetics of this, including twin studies. Those studies have basically found that the heritability of OCD is somewhere between 20 to 35% or something like that, maybe 40%. This is the largest study to date using a technique called GWAS, which is basically a way to look at single nucleotide variations across the genome. And what these investigators found was a variety of genes that are associated with the risk to develop OCD. But their primary focus was to understand whether they could find genes that were less common that were also underlying OCD, because previous work found that it was only very, very common genes that seemed to be related to OCD and not rare or less common genes or less common variation in genes. And so what these investigators did with this larger sample was basically to demonstrate that the heritability component of OCD, which was about 30% in this sample, is constituted by genes that are both common from the standpoint of their SNP variation, and also SNPs that are relatively uncommon, having a allele frequency of less than 0.05, for example. So what's interesting about this is that this sort of changes the way we look at the genetics of OCD. Prior to this, people saw this as being somewhat different than other psychiatric disorders from the standpoint of not seeming to implicate genetic variation that was rarer. But now this study demonstrates that, like other psychiatric disorders, the heritability at a genetic level is complex, that there are many genes that are involved in contributing to the heritability, and that this is across the rarity versus common spectrum. If there's a SNP or a genetic variation that's rare or common, both can be involved and are involved, it looks like, in underlying this risk. So this provides some insights into not only the genetic underpinnings, but ideas about thinking about perhaps how we move forward in further understanding the pathophysiology of OCD. Dr. James Leckman from Yale, who's an expert in the area and a longtime researcher, writes a really nice editorial about these data, putting them in context of sort of genetic variation, but also talks about environmental factors, which are also highly relevant to all of our psychiatric illnesses, including OCD. And so it's really, I think, valuable to check in on the editorial that Dr. Leckman wrote as well. The paper by Weinberger and colleagues from the Lieber Institute is, I think, quite exciting and is focused on understanding the genetics that are associated with suicides, but in this particular case, a focus on violent suicides. The work that is done by this research group uses data that has been collected from postmortem brains in a brain collection at the Lieber Institute in Maryland. And in this particular set of samples, the investigators were able to investigate, to look at gene expression changes in a region of the brain that's critical for emotion regulation and for the interface between how we think about things and how we feel about things, and how we think about the future and combine thinking about the future and planning to solve problems with our emotional lives. And this is an area of the brain called the dorsolateral prefrontal cortex. Its alterations in the structure and function of this region have been implicated in a variety of psychiatric illnesses, including schizophrenia. What these investigators did was to look at gene expression, which is the product of the genetic variation and also any environmental influences on our genes, epigenetic phenomena, to understand gene expression changes that may be associated with suicide, and in particular, violent suicide. So in this particular case, what the investigators do is they sequence the RNA, and again, the RNA is the product of not only the structural variation in the genes that we inherit, but also how our genetic genes work in relation to the experiences that we have in our world. And what they found is that there's a specific genetic signature that is associated with violent suicides that differentiates individuals who have killed themselves by violent means from individuals that have committed suicide by non-violent means. So, for example, a gunshot wound versus a drug overdose. This is really, I think, potentially very, very interesting. There's a certain set of genes that were identified, and the kinds of genes that were identified to be specifically associated with violent suicides were related to a purinergic pathway, which is quite interesting because purines are molecules in our bodies and our brains that we, most of us think about in relation to the backbone of DNA structure, but also are very much involved with neurotransmission and signaling and the modulation of neuronal and glial function. And so what these investigators found was not only were these genes and their expression patterns associated with violent suicides, but also they significantly differed from individuals that either did not die by a violent suicide, but died by a non-violent suicide, or other non-suicidal patients, and from controls as well, to some extent. This is new work, it's groundbreaking. It suggests that violent suicides may have their own sort of underlying genetics and pathophysiology. And also, one of the interesting findings was that regardless of diagnosis, these findings held up if an individual committed suicide by violent means. The genetic findings were not determined by whether you were schizophrenic or bipolar or had major depression, but rather this finding of the alteration in the expression of genes related to the purinergic system seems to be relatively specific for violent suicides. This really has implications also, I think, for thinking about, as I mentioned, diagnosis and potentially down the road, even treatment. And then finally, we have a paper on autism spectrum disorder, looking at the heterogeneity of autism spectrum disorder in relation to cortical thickness and gene expression data. There's enormous clinical heterogeneity across individuals with the diagnosis of autism spectrum disorder. Folks that treat individuals with ASD are well aware of this. This is a study that was done to try to understand whether or not we can learn more about the brain changes that are associated with some of this heterogeneity in the presentation of symptoms, and also to understand whether or not there are different genes that might be associated with these different presentations of symptoms and patterns of brain alterations. And to make a long story short, researchers identified in a group of 360 individuals with autism spectrum disorder, as compared to roughly 280 controls, that there are changes in cortical thickness in individuals with autism spectrum disorder. This has been well known. They found reductions in cortical thickness in some areas and increases in other areas of the brain as compared to controls. Areas of the brain in which there were increases included such regions as the anterior cingulate cortex and the anterior temporal lobes. But one of the interesting things that these researchers also did was to look at brain regions in which they did not find differences between the groups, but found that there were more individuals with ASD that had a lot higher or a lot lower values than control individuals. In other words, looking across the brain for individuals that were outliers in their cortical thickness measures. And so what they did is they found regions that were not on average different between the groups, but regions that had more ASD individuals that had very high or very low types of values. And by doing so, they identified numerous other brain regions that appear to be affected in individuals with Alzheimer's disease from the standpoint of having greater variation in cortical thickness. And they were also able to show that that type of a measure was somewhat predictive of the likelihood of having ASD, as well as the type of symptoms that one would have, and that this also was related to a measure called our polygenic risk score, such that by looking at all of the genes that are maybe involved in ASD, that is predictive of these individual differences in measures of cortical thickness as it relates to measurement across the brain in these regions where they're outliers or where it's atypical. So it's a little bit complicated in thinking about the analysis, but again, a very important step from the standpoint of thinking about how to look at structural brain alterations across individuals that have ASD, relating those alterations to symptoms as well as those alterations to genetic variation. So in summary, what I think is really important to keep in mind about this issue is that it really is our, I think, demonstration in the field of the advancements that we're beginning to make in relation to genetic vulnerabilities as they influence the development and course of psychiatric illnesses. And these are, four specific examples related to, in one case, a mutation that is significant and, is related to neurodevelopmental disorders, but also psychosis, which provides insights into illnesses like schizophrenia, furthering our understanding of the genetic architecture of OCD and realizing that its genetic architecture is quite similar in many respects to other psychiatric illnesses from the standpoint of its complexity. Really understanding and breaking new ground, I would say, in relation to this idea about violent suicides as being a distinct category that runs across diagnoses, transdiagnostic, if you will, with genes that are altered in the way that they're being expressed in the dorsolateral prefrontal cortex of individuals that are specifically related to purinergic signaling. And then finally, using phenotyping and behavior and symptoms as well as cortical brain alterations and polygenetic risk scores to better parse the heterogeneity related to ASD. So it's an exciting issue, and I think these papers, along with the reviews that we provide, should be something that will be of benefit to lots of our readership.
Dr. Kalin, thank you once again for putting the issue into context for us.
You're welcome.
That's all for this month's episode of AJP Audio, but APA Publishing has other podcasts you can listen to. From Pages to Practice looks at the latest research published in the journal Psychiatric Services, hosted by Dr. Lisa Dixon, editor-in-chief of the journal, along with Dr. Josh Berzin. Psychiatry Unbound is the books podcast from APA Publishing, hosted by Dr. Laura Roberts, editor-in-chief of APA Books. Check out these and others, along with APA's newest podcast, Mentally Healthy Nation, at psychiatryonline.org/podcasts, or you can subscribe via Apple, Google, Spotify, or wherever you find podcasts.